DNA is very finicky and like the process is not like optimized and a lot can go wrong. So it depends on your research objectives. Like for example, even the primer, like what you decide to use as primer, it depends on the goal of your study. Because there are a lot of different primers for a lot of like different reasons to use.
Thank you.
So that means that there's still a lot to learn. More research.
Thank you, Monica. Laurence, you want to start? We have some minutes for short questions.
Okay, a short question. You explained that you did 2 sampling campaigns in your MDA. Were you able to see if there were some differences from the focus sample that go from April and the one that was from fall? Try to be fast.
Yes, I wanted to show it like on the facet graph, but I couldn't. It's not here. But so we see like because I primarily did the 2 sampling sites to compare like if there's a difference between the species composition during winter or cold months and during April, which is like hot or summer months. So there's a bit of difference, but not as much as difference compared to the effect of salinity. So mostly they're the same, like, but because it's like subtropical, so we assume that there's not really like a huge difference in the species composition. So species composition largely differs due to salinity, but not due to temperature.
Which means that the fish do not travel along the river according to the
temperature, yeah, but according to salinity.
Okay.
Thank you, Laurence. Luana?
Yeah, thank you, Jas. Mariska,
Yes.
congratulations.
Thank you.
We're really happy to see even the preliminary results of your thesis. They're very, very interesting. So I'm looking forward for the final results. And yes, that's a very interesting oceanographic work. You do a connectivity from between salinity, fresh water to salinity environment. So I think you produce a very important key piece of work. There's a long way to go to achieve the major goals overall, and the goals of your thesis you are achieving the major goal that you lastly presented on your introduction. What are the next steps?
So next steps, I need to include one human factor since we're associating the tourism activity to the species composition, and then like make a better because one of the problems as well for eDNA is for normalizing the data set that we have, since we have like huge difference between the raw reads between samples, which I didn't show because it's another conversation to have. So we need to make a more robust analysis and identify which, not really correct, but which process is more apt for the data that we have right now, since that is still like preliminary.
And when you conclude it, what do you think it will be done next with the data that you are producing? Or what do you hope that will be done with it?
So first is since the main goal is for identifying the fish species, is to confirm that what we annotated is really like the correct annotation. We will do that by confirming it with the expert from Kagoshima University Museum. And then the next one is to make sure that the data analysis that we did for our sample is correct, and all the results that we have, like our graphs, our NMDS, and all the correlations that we do are correct based on the correct bioinformatic pipeline.
Okay, thank you.
So to do. We didn't want
Thank you.
Good. Okay, so thanks a lot for such a nice work. So I really enjoy it. I think even though it's preliminary, you show many things, so it's great. It's not so preliminary. So I have more methodological questions. So did you use cocktail primers for?
I used my fish 12S.
Yeah, but only one pair of primers, or?
You separate the PCR, or you put in the PCR, the first PCR, you put everything together?
Yes. So primer and.
But one pair of primers, not a cocktail. And you put two primers or 2 pair of primers? 2 pair,
like forward and reverse.
Okay, not one, so 1 pair, not a cocktail. Because in eDNA, what you might have is some assignment to certain species might depend how well those primers are in, and you can compensate by that, because that could make that one species amplify better than other. So by applying the cocktail, several pair of primers, you might compensate that. That was just mythological, was just to be sure.
But we did something similar, which I didn't include because that part of the experiment did not really push through. So we used two different enzymes. Instead of like primer cocktail, we used kappa and prime star. And then some species are amplified in prime star, some are not amplified in kappa. So the results that I showed are from kappa only.
Okay, and just which is the length of the size that you use? The length of for sequencing? Which is the length?
350, 370, 350.
350?
Uh-huh.
Okay, and you
With
couldn't
all the
with adapters and everything. Okay, so maybe it's like what, 200 something?
Yeah.
And there's no difference between Niloticus and
so it's 100% match. The one that I have for
that was,
the blast
I
is
was surprised by that,
160, yeah.
because I mean it's CO you're using on which one is 12. But I mean in 300 or 200 base
The
pairs,
sequence that I run in NCBI for Niloticus is like 165.
and there was no difference, it was identical.
It's 100% match.
Yeah, I mean
So-
some part is less variable than others. Okay, it's fine. And the other thing, the last one I promise. So, and it's more about mythological aspect. So why did you -20 directly instead of ethanol? Which was your decision about that?
Because we had like quite some time before we did the extraction. So we added DNA iso and then put it in the freezer because it's more stable that way.
Okay. Okay, not directly in ethanol.
No. So
Okay.
after filtering in Sterivex, which takes a lot of strength,
Yeah.
we add DNA iso and then
Okay. Okay. So you keep like okay. It's a handle.
yes. Thank you very much. You have questions?
No, no one. I have many questions, but we can take a coffee.
Yes.
No one. Okay. Okay, so now we have Akshaya. I pronounce it well?
Akshay.
Okay.
Hello everyone, I'm here to present my thesis on the neuromodulation in the visual processing system of Octopus vulgaris. I was supervised by Dr. Giovanna Fonte at the Stagione Zoologica Anton in Naples. No!
That's the more important one.
Okay. Octopus vulgaris, as we know, they are found in the coastal waters and they are benthic species. They are abundantly found in the Mediterranean Sea, and we focused on the Bay of Naples, Italy. Octopuses, along with squids, cuttlefish, and nautilus, they form the class of highly intelligent marine invertebrates called the cephalopods. And they are also highly studied species among the group. But why do we need to study the octopus? Because they possess around 500 million neurons, rivaling that of the vertebrates, and their nervous system is advanced, which possess neurons capable of learning and memory, which further allows the octopus to express a variety of behaviors. And despite evolving in parallel with vertebrates, they seem to possess convergent cognitive abilities after they lost their external hard shell around 530 million years ago. And they're also of high economic demand and in general for human interest. So the purpose of my research is to help our understanding in the visual processing system of Octopus vulgaris, especially to characterize the neuromodulator molecular fingerprint, because neuromodulation studies are essential to understand the neural basis of why the octopus behaviors in a certain way. But before we talk about the neuromodulation, we need to understand the anatomy of the octopus brain. So the nervous system is present between the eyes of the octopus, with the optic lobes present just behind the eyes. And they have a central nervous system which possesses a high degree of centralization and consists of two lobes, the supraesophageal mass present dorsally on the to the esophagus, and the subesophageal mass present ventrally to the esophagus. So it's like around the esophagus, and they have 8 nerve cords which innervate the 8 arms. And why we focus on optic lobe is that they are the main center for processing the visual sensory cues and also aid in the storage of visual information. They also have a peduncle lobe complex outside the, just outside the optic lobe, which is responsible for the control of the visual motor sense cues. They possess an external double layer which consists of further 3 layers like the outer granular layer. One thing I'd like to point out is that this layer is also known as the deep retina because the anatomy of this layer is very similar to that of the vertebrate retina. And then the plexiform zone, which is a neuropile zone, it does not possess the neurons, but then all the processes are present, and then the inner granular layer, followed by a region which consists of the region called the medulla, which has a lot of cell islands, which are then surrounded by neuropile tracts. And then the peduncle lobe complex, which also includes the olfactory lobe and the optic gland along with the peduncle lobe. And the peduncle lobe is this one here, and it's very close to the optic lobe. And why do we need to study neuromodulators? Because they act as tools of neuronal communication, they influence the signal transmission, and because of which they are important in studying this synaptic plasticity, which aids in the neural adaptation of the organism and enhancing its ability to learn and remember, which further influences the various behaviors that it shows. Neuromodulators and neurotransmitters have often been used interchangeably, but then the key difference here lies in the mechanism. Neurotransmitters here, they release the chemicals which are transmitted from synapse to synapse through ion-gated channels or G-protein coupled receptor pathways. Whereas the neuromodulators, they release the chemicals in the extracellular space so that it can target multiple neurons at once. For this research, we chose 9 neuromodulators like dopamine, octopamine, norepinephrine, glycine, GABA, etc. And we also chose 9 genes which corresponds to the respective receptor or transporter genes. So we chose the receptor genes for dopamine, octopamine, norepinephrine, serotonin, and histamine, whereas we chose the receptor, the transporter genes for the rest of the neuromodulators. For the methodology, we collected the optic lobes from the animals caught in the Bay of Naples, and we processed them. We sectioned them using a vibratome, and further we used in situ hybridization technique to localize the specific mRNA targets. Further which, we counterstain the tissue with DAPIs so we can study the cellular localization and the organization of the neuromodulators. And we used dark field microscopy to visualize them, and further we process the images using ImageJ software to So we categorized the results into the regions, double layer, medulla, and the peduncle lobe. So in the double layer here, the image A shows it's the representative image of the whole slice of the optic lobe, sectioned coronally, and you can see that these rows of images were acquired in these particular regions, and you can see that the outer granular layer, the plexiform zone, the inner granular layer, all they're pretty clear. And so we found that for the outer granular layer, we found that the genes TH, TBH, GLIT, and these genes to be more pronounced, whereas we found the DBH was more pronounced in the plexiform zone. And for inner granular layer, we found most of the genes present there. For example, I would like to show the organization of these two genes. We can see that the different cells expressing the different the genes, and you can see the patterns are very different and distinct. The sizes and the morphologies you can see. Similarly for medulla, if you see the three regions in the medulla, you can see that there are numerous cell islands, again of various sizes and numbers and different shapes. And you can see that the DBH is not present in the medulla, whereas you can see few cells of TBH, HDC, VAC. You can see the fine localization of this gene. And for example, I would like to show the VAC with the merged image of all the fluorescent image channels, with specifically of VAC. And you can see that you can compare that these cells are actually expressing the gene VAC by looking at it with the DAPI counter stain, because the DAPI shows the nuclei. So you can see that the cytoplasmic expression of VAC. Similarly, for the peduncle lobe complex, we can see that DBH is not present here as well. You can see the few cells in the peduncle lobe showing TH, and a specific population of cells expressing DBH. And you can again see that very few cells are expressing TBH, and numerous cells expressing the GLIT genes. And further, you can see the different distinct populations of cells expressing the VAC, HDC, and the VGU genes. And this is a zoom in of the gene HDC, for example. And further, we summarize the results in the optic lobe to show the overview of the spatial organization of all the neuromodulators present. And you can see that in the double layer, or specifically the outer granular layer, you can see that the octopamine, dopamine, norepinephrine, glycine, and glutamate being present, whereas the rest of the neuromodulators are absent. And in the plexiform zone, you can see that only norepinephrine is expressing, and in the medulla, norepinephrine is absent, whereas all the other neuromodulators are present. So we try to compare the possible functions with the vertebrate visual system, and we know that dopamine, serotonin, and acetylcholine, they're important for learning and memory functions. And here with as I said before, that the medulla is responsible, that the medulla is the center for the memory storage for the process of the visual information. We can say that it's responsible in sensory perception, and dopamine was found in throughout the optic lobe. So they are responsible for a lot of the learning and the memory functions, and that the optic lobe has dopaminergic cells present throughout. Interestingly, for acetylcholine, a study was done specifically to localize the cholinergic cells, and they found that it was present in the inner granular layer and the medulla region, same as what we found here in this research. So we can also say that acetylcholine is indeed involved in various learning and memory mechanisms. Norepinephrine is supposed to be absent in invertebrates because octopamine performs the functions analogously. However, here in octopus optic lobe, we found cells that expresses the norepinephrine neuromodulator. GABA and glutamate and glycine, they are important neurotransmitters in signal transmission in the vertebrate retina, and we found them to be present in the octopus optic lobe cells as well. So we can say that they could be involved in these processes. Histamine has its role is still undefined in the vertebrate retina, but then it is an important photoreceptor transmitter in insects and crustaceans. However, the data is limited, so perhaps they could have a role in the octopus vision as well. In conclusion, we were able to localize and identify this populations of cells expressing the different neuromodulators and involved in the different processes. Thus, we contributed to helping fill the knowledge gaps with respect to the octopus vulgaris nervous system. And future studies with advanced techniques like electron microscopy could help achieve a final localization of the neuromodulators. So we can understand the functional link between the neural basis of their complex behavioral processes. So further we can understand why, how their behaviors are working. And finally, I would like to acknowledge my supervisors and the research institute that I did my thesis at, and all my lab mates. They have been incredibly supportive and encouraging in this pretty tough topic. I still like octopus. And thank you all.
Beautiful pictures. A question from the audience. Anyone want to know something? No? Okay, so it's your turn now. Yes.
Okay, thank you guys. Congratulations, thank you. Very interesting work. Yeah, it's not for everybody to do, but I'm really interested into animal behavior. So I don't know if you had thought, because my question is more in a broader sense of what your research, the results. I think it's very clear the relevance of what you achieved with this research. But did you, while you studying for it and to make your discussion, had any insights on two aspects? One, do you expect to have differences with other octopus species? And the other is, do you expect that their modular system can be affected depending on where they live? What are their environmental interactions that would eventually affect their behavior?
So octopuses are solitary species. So and recently, especially with the studies that my supervisor has been doing. It has been they have been observing that octopuses they have inter-individual differences. So there are so many differences even with individual octopus vulgaris species. So and neuromodulation studies are pretty recent. So there could be future studies.
And then you could expect that
Yeah.
other species would be different.
Yes.
And then could but do you have any insights on how you could apply this knowledge on a more ecological approach over the species behavior?
No, it was very neuro biology,
Yeah, I know.
morphology.
It's a lot of time. But what would you, let's say you have the chance to continue this line of research, what would you like to investigate further and that you think it will be relevant?
Actually, the semester I had in Oviedo with Gonzalo, professor, the population dynamics. I think it's a very interesting area as well. So maybe combine this with population studies, because like octopus vulgaris is found throughout the coastal waters, the tropical and the temperate. So then we could have interesting results in those studies.
Anyone who's one question?
Oh, wow.
Do you? Yeah. Yeah. But I think I forgot my question. I don't know. I remember. What is your opinion concerning ethical approvals and how we interact with the species? In the sense we don't really interact, but how humans are using as a resource octopus, knowing that they have such a
There are so
complex
many angles to
neurological
this.
system.
What is your opinion?
What is your opinion? I want to know, what do you think about it?
So the thing is, with these experiments, you need a lot of animals. Even with optic lobes, it's just 2 per animal, so you need to keep catching the animals. And people like to eat octopus, so
But I like it too, but it's a questioning that I have. So you are
Yeah.
assigned this validated research,
yes.
but just would like to hear what you, as the science about to be, what is your opinion on this topic?
Well, as a scientist, I would like to study more about octopus, but at the same time, the ethics are in question, because I wouldn't want to work with octopus if I'm the one having to anesthetize it and further.
Respect the protocols. I mean, it's just
Yes.
respecting the protocols. Everything can be done if you respect it.
Yes, we can follow the ethical protocols,
That's it.
definitely, but it's still a moral conscience
Yeah.
at the end of the day. So.
But maybe it's just a hint at what's more the research is needed,
Yes.
so we can find out.
Yes.
But should we still be exploring the resource the way that we are exploring? That's just an open question.
Yeah.
Thank you.
Well,
Thank
it's
you. Good to learn.
Okay, go.
So thanks a lot for such a nice presentation.
Thank you.
And I mean, I've never seen such an amazing topic. So it's amazing what you
Thank
did. And I hope that if you did that, it's unbelievable. I mean,
This is still not so clear. With the confocal microscope, it would be clearer,
but it's really nice work. I think the characterization is really good, so great. The only thing, I would start in a different way. So I would start, when you say make future thing, I would say at the beginning, like,
Okay.
we have this knowledge gap.
yes.
What I'm going to do is to prepare the basis for doing this. Because that's what you're doing. Because
Yes.
it was, before this study, nothing was done about this. So for example, just came to my mind, I have no idea what this.
Okay.
First of all, I'm not an expert now. But which is the effect, for example, for mating? Yeah, for color, or whenever some color change is involved, how that is, and you are putting the basis for anyone later in the future to come to that. So, and I think you need to to make it even, you're making the basis for that. So it's an awesome contribution, and I think it's this kind of. It's true that at some point it could be a little bit descriptive, but I think it's this kind of study are super necessary. I mean, and the other thing is like it would be nice. I mean, depending on when you analyze the evolution of this is more like a reflection than a question. But when you analyze the vision and how different genes are involved and receptor and neurotransmitter, so how that's evolved because it's octopus there in some particular lineage that it's interesting to see how this neural development works in comparison with other lineages. So I think it's so I don't have questions. It's just a reflection and people should really appreciate what you did and so well done. Thanks a lot.
Thank you.
Thank you. And Laurent?
Just a quick question that is related, in fact, to what he said. You presented the specialization of all those cells in the different structure of this part of the lobes. I was wondering if you have an idea, like if in others or close species, if the same structure, when the same structure exists, if the same group of cells at the same location have lost or have specialized in the same genes, or if this work of those that are not existing.
Before this, there was a study that was done, but in octopus embryos for the neurogenesis, but so far it's not in any other species except octopus.
Okay.
There is also another study that characterized the neuromodulators in octopus bimaculoids, but not other than that.
Okay. Well, thanks, because it's I'm not a specialist, so I see all those as cells specialization, which is very, for me, it's surprising. So I wanted to know if it would be common in other structure. Okay, well, thanks for your answer.
Thank you.
Okay, I have very fast questions. What you were showing to us were genes or proteins?
Genes.
Again,
mRNA.
what you're looking in your picture is the expression of the genes. No? You are marking the receptors, or no? Correct me if I'm wrong, but the genes are in the DNA of all the cells.
Yeah.
The same genes. Another thing is if that gene is present or not and where. So what you are observing is the expression of the gene.
Yes.
Not exactly the gene. I mean, it's the protein, the receptor, etc. That's the protein. Okay, it's just a detail, but it's important because I mean all the cells have the same genes. Another thing is in which tissues and in which if you are expressing that genes. It's another thing. Another thing I would like to know is if you want comment very briefly how was the process to select the genes? To select that 9 genes. You participate in that discussion with your supervisor, or it was? I mean, because I have been directing thesis, and sometimes you arrive and you need to do this, and there is no time to make discussion. But maybe you have the opportunity to think about why those 9 genes.
So, my thesis research is an extension of a postdoc researcher's PhD project. So she characterized them using informatics, data analysis, bioinformatics. She added a lot of genes, and we just selected these because we have the probes for these.
Okay. Just to finish, to say that behavior, or to try to understand how is the behavior of octopus, the reactions, and how physiologically is responding, is very important for conservation and sustainable management of the species. Because, for example, in Asturias, I mean, you were in Asturias in Oviedo, and there is a sustainable fishery of octopus. And we were making genetic population analysis with Negin, that was one student from this master, about that. And it's important, and the octopus is something very surprising because, for example, you have a big population and you say, okay, next generation we will have a big population. No, there is cannibalism and they eat each other, and the next year you have a very small population. It's like this. It's crazy. So, let me congratulate you. Say that your 15 minutes are over, so you are master in science, and say I congratulate also your supervisors. Okay? Thank you so much. Okay, and the last but not least, Luna. Okay, floor is yours.
Thank you very much. So, good afternoon, everyone. My name is Luna Baudron, and I did my master thesis in São Sebastião, Brazil, on the spatial temporal pattern of free fish community productivity in the southwestern Atlantic, supervised by Vicente Pineiro in Sibimar.
It's like this.
Too much of it.
Calma.
Don't worry. No, if you don't click just picture like this. No.
I try to do something. Don't worry.
No.
We can try to do it like
Any other way?
Because it's idiotic.
So
It's like
Up like that, like this. Try like this. Up.
Okay,
And
let's start again.
yeah.
Okay, start again.
Start again, the word.
Okay. So, good afternoon again. My name is Luna Baudron, and I still did my master thesis in São Sebastião, Brazil, on the spatial temporal pattern of free fish community productivity in the southwestern Atlantic. What a surprise! Supervised by Vicente Pineiro in Sibimar. So, as you may know, coral reefs are important ecosystem for local communities for various reasons. For example, food and resources security, economical income and employment, coral reef, coastal protection, and social cultural services. Unfortunately, these ecosystems are highly threatened by a large-scale climate change and local-scale anthropogenic pressure. In order to implement adapted conservation measures, a better comprehension of the community structure and function is required. In this context, fish productivity has been created. Fish productivity is a great tool in order to look at which areas are more sustainable. Fish productivity can be defined as the sum of all individual gain of mass per day and per hectare. This measure integrates species-specific traits in the calculation of biomass in order to replace each individual in their growth curve. What does it mean concretely? A fish of 10 cm will grow differently depending on whether it's a juvenile that can reach 30 cm or an adult that already reached its maximum size. So, this measures integrate all those parameters in order to have a better comprehension of the ecosystem drivers and dynamic and processes.
Yes.
So this measure had already been used in Indo-Pacific, where in this region the productivity can be predicted by pelagic subsidies and associated with a large amount of plantivores. In this region, the ecosystem is slightly different. It's more characterized by abundant and diverse branching coral in more oligotrophic water, while southwestern Atlantic is more characterized by massive corals in turbid, deep, and nutrient-rich water. Those characteristics may have an impact on the productivity. The knowledge on marginal province like southwestern Atlantic remain limited. That's why the aim of this thesis is to identify the main predictors of the fish productivity and the associated trophic yields in the southwestern Atlantic. Our main hypothesis is that this ecosystem is driven by pelagic subsidies and associated with plantivores. In order to validate or not our hypothesis, we use generalized additive model. This model allows us to see non-linear relation between the predictor variables, in our case environmental data, and a response variable, productivity. This process follows 3 steps. First, we're going to calculate the productivity, then we collect the environmental data, and finally, we associate them into a generalized additive model. So, for the first step, the productivity calculation, we need data from the field and theoretical data. Let's have a look to the first one. Data were collected in Alcatraz's archipelago, a marine protected area located 35 kilometers away from the coast of San Sebastian. Since 2020, 3 sites were annually monitored. Those sites have different characteristics, but what is important is that Alcatraz's is actually really representative from the entire coast of Brazil. So, underwater visual sensors consist in a single diver looking within a 20-meter transect and 2 meters on each side. This diver will estimate the species and size of each individual encountered. It's a huge job, but they managed to do it well. On top of that, they have to also estimate the abundance when it's a large group. So then, we look at theoretical data. For each species encountered on the field, we look into open access database for the specific characteristics as a maximum length or diet. Then, we associate each individual found on each transect to their own characteristics, as for example in this example table. Now we are ready to calculate the productivity on R thanks to the package RFishProd developed by Renato Moraes. This process also follows a few steps. The first one is the calculation of Kmax, which is a coefficient of the von Peterlofik growth model, which allows us to calculate the weight of an individual at day 0 and the gain of weight at day 1. Then, thanks to the Gilasson model, we estimate the mortality probability and we remove stochastically some individuals based on it. Finally, the productivity is just the sum of the remaining gain of mass. Then let's go to the second step, the environmental data collection. So we need first benthic coverage. Those data were also collected in Alcatrazes on the 3 sites mentioned before since 2020. For this collection, the diver will look exactly under the transected which organism is present or not. Then it will be categorized as one of the 8 categories present here, and we will estimate it every 50 centimeter of a 15-meter transect. Then, at the end of each transect, an average coverage of each category is calculated. Then, we want annual and seasonal chlorophyll A concentration and sea surface temperature. For this, we collect them thanks to the satellite Aqua MODIS from NASA on the monthly average of available data. So now we are ready for the third step. On top of all the environmental data collected before, we add the predictor variables year, site, and species richness. The best model will be chosen based on the lowest AIC and the principle of parsimony. So here are the results. The productivity was found to be 3.329 kg per hectare and per day, and the main predictors are bare rock, coral, and turf coverage. We can see a positive relation with coral cover and a negative one with bare rock and turf. So then we look more at who drives the productivity, which type of trophic gills participate to that. So we did some exploratory analysis, and we found that 75% of the productivity is due to only 10 species. And even more impressive, more than half of the productivity is due to only 3 of them: Ireland or Lineatum, Abodef saxatilis, and Kipos sp. So, if we look more at the distribution within the 7 trophic gills, we can see that invertebrate mobile feeders contribute to a large amount of the abundance and the productivity, followed by omnivores and fish and cephalopod feeders. I want to catch your attention on the difference that exists between abundance and productivity. For example, omnivores and planktivores have approximately the same abundance, but have a way different contribution to the productivity. So our analysis shows spatial then we did some other generalized additive model for each trophic gills. So here are the results for the main one, the invertebrate mobile feeders. And we can see a negative relation with seasonal chlorophyll a positive one with canopy and coral cover. Canopy was found to be inverse covariate of turf. So that means that canopy and coral coverage corroborate the results found on the general model. So our analysis showed that the relation between fish community structure and benthic coverage is really complex and really interesting. In the southwestern Atlantic, the environment and cover diversity and abundance is different than the one found in Indo-Pacific with much more branching coral. But the productivity is still found to be approximately the same as the one in Indo-Pacific, except from the hotspot of fragile plant. But which is interesting is that the main trophic gills is different. In the southwestern Atlantic, it's found to be invertebrate mobile feeders and omnivores, while in Indo-Pacific, more than 40% of the productivity is due to planktivores. And this reveals an ecological dynamic which is different. In southwestern Atlantic, plankton represents a large source of resources which is underexploited by specialist fish as planktivores. Instead, more generalistic and opportunistic feeders as Iron or Lineatum or Abodef saxatilis feed on this empty niche. And the productivity of those 2 species are really important for the general productivity. So our analysis revealed that the spatiotemporal variations are mainly due to benthic attributes, as the negative relation with bare rock and turf and the positive with coral. Oh.
Up. No, you want to close that, no? Okay, once.
Sorry. Okay.
Or is it left for me?
Okay.
Oh, well.
Don't worry, we will give you the time. If I do not do it, one more?
Yeah, back. Okay.
You're going back in 5.
Yeah, it's where I wanted to go. So yeah, sorry. with seasonal chlorophyll and the main trophic gills, invertebrate mobile feeders, those results tell us that the quality of the habitat is an important factor for predicting the productivity. So if we look more into each result, the increase in coral cover is associated with an increase in biodiversity, complexity, shelf and food resources for many organisms. And once the most important fish species are generalistic that feed on adjacent salt bottom and water column, those productivity has a great impact on coral cover because they bring nutrients from the outside of the coral reef and have a great impact on the coral cover and consequently the coral reef. Then the relation with the benthing and pelagic producers, respectively turf and seasonal chlorophyll, suggests another ecological dynamic. Herbivores and omnivores, although not the most abundant, has a great impact on the productivity, and this important can lead to a top-down control on the turf coverage. On the other hand, plankton represents a large source of resources consumed by zooplankton, highly abundant in the region, then consumed by generalistic and opportunistic. This leads more to a trophic cascade effect. So, in conclusion, productivity in the southwestern Atlantic can be predicted by benthing attributes, coral, turf, and barrel coverage, and associated with invertebrate mobile feeders and omnivores. Which is striking difference with Indo-Pacific, which is more predicted by pelagic subsidies and planktivores. The relation with coral is due to external nutrients brought by generalistic feeders that have a great impact on coral coverage and then coral reef, while the negative relation with turf coverage is due to a top-down control of herbivores and omnivores. So I want you to remember the importance of the habitat quality for fish productivity and consequently local communities. Fish productivity is a great tool for better management of area, for fishing, for management, conservation in general. Thank you for your attention.
Thank you so much, Lorna.
Thank you very
Any
much.
questions from the audience?
I have a question.
Okay.
Hi, I have a question. Could you go back to the slide where the sampling was conducted?
Sure.
Uh,
Are you sure you want to go that far?
I just want to clarify if I understood correctly.
Sure. Wait
Yes,
just a
yes,
second.
Nice one.
The
The
coverage?
Yeah.
This one? Or the
with the
fish
fish.
one?
Okay.
Ah, okay, sorry.
Sorry, the fish. There's that too.
Here,
Yeah,
yes.
no one. My question is because I'm not a coral reef or fish person. Just to clarify if I understood correctly, you did the sampling to identify the species or individual fishes through visual.
Yes, to visual and water senses, which means the diver will be in the water and just look what he can see on the range of like 2 meters on each side to see. Actually, it's a bit more complex than just this shape. It's just like on the way in, he will estimate the fish on the water column mainly and the shy ones, the one that can escape, to be sure that to count them and to register them.
But my question with that approach is there any way when you analyze your data that could avoid bias when it comes to the visualization of the fish? For example, if there's a clownfish that the diver will see for the first time and then will do it again, what is the chance that it's the same clownfish and not a different clownfish to say that there's a lot of the abundance of that specific fish in the sampling area?
Because the diver is like is going on one way, so he will not swim faster as fish, of course, but he will consider that he see the fish once, because the fish usually try to escape.
Swim away.
Yeah, escape, swim away. So normally, it's rare that a fish come back, or if he come back, that means he's like attracted to you and he will just stay around, you know. So yeah, no. If you want, it could be a possibility to do like with cameras and analyze the video after, but actually it's not really productive because you have to, at the end, do the job twice because you have to go through all the videos again, and it's a pretty great procedure. Like it works well, and the divers are trained before and trained with people that already done that and homogenize the result. For example, if there is a big school of fish of 200 fish, okay, someone can say, oh, that's 200, and someone else that is new will say, oh no, it's 500. It can be avoided by previous training, and so, of course, there is bias, as always, as we're in science, nothing can be perfect, but I'm pretty confident of this.
Yeah. And can I have one last final question?
Very short.
Yeah, sorry, really interesting though. How about predation? Because you have a set of the, let's say, the abiotic factors that could relate to the productivity. Is there any literature that says that in a particular area, especially if it's really pristine or covered, is predation a big factor for you to calculate the productivity? Let's say it's very productive because there's no predators or diseases, for example.
So, the predation will be integrated more on the trophic gills, fish and cephalopod feeders, so carnivores, and it's a limitation of the calculation of productivity in the sense that the mortality probability that we calculate into the productivity calculation doesn't integrate the predation. So this is still a tool that can be improved in this sense.
I see. Thank you.
Thank you.
Thank you very much.
Thank you, Gonzalo.
Yeah, thanks. So, congratulations. I think you did great presentation. I really like your pace in the presentation. We keep attention all the time, so well done in that sense. So yeah, I also have several questions about this because I think it's really interesting. At some point you talk like in future tense, but you did this, no?
I did.
So you have the data.
I have the data.
Okay, yeah. So my question is, can you show the repeatability of that? Let's say if you took 2 people or 3 people, because normally when you do this kind of survey, I try to do it. I'm not a perfect, really good diver, but I did some of those, and you have variation.
Yes.
And you have variation between divers.
Yeah.
And in fact, you correct by that by doing both divers going more or less to the same area and see which is the maximum and the minimum for that. Because if not, if you go to different regions, what you need to have is the same observers all the time to have some sort of the same bias. Have you tested that repeatability that you're having?
So
Because correct me if I'm wrong, but would you? I mean, I know that this is simplification because there's some models that show clearly how with the distance, so the confidence distance is 2 meters because it's not the reliability or the feasibility is dropping down. This is well known, this kind of survey.
Yeah.
That's your question. But you estimate in size? No. Just
yeah, we
present
estimate size also.
by eye?
By eye, yes.
Yeah.
But then within certain categories, let's say this
Yeah,
category,
yeah.
this. So how can you show me the categories that you? Because I'm sure that you're gonna say, oh, 51 or 38. So it should be within certain categories, no?
It's within certain categories, for example.
Maybe like a range.
Like a range, like a 50 to 55.
Okay.
And on my
okay.
calculation, I needed a precise number. So I took for example, if it was between 50 and 55, I did 52.5.
Yeah, because I mean it's tricky that these 30 as an example, this 38 centimeter should be a range, not really a 38 centimeter. No, correct me if I'm wrong.
On the productivity, I cannot. On the R code, I need to have a specific number.
So you put the mean of the range.
Well, exactly.
Okay.
Yeah. So, but yeah, I
Did you
think
test for repeatability that you have for certain sites or whatever?
so. As I said, there is a training between the divers that
No,
do
no,
it
but
in the
apart
summer.
of the training,
Yes,
once they are
and-
trained,
Yeah.
have you tested repeatability at least in one point or whatever to see how many times you get more or less the same estimate
Yeah.
in one sampling point at one time? That's my question.
Actually, I said that it's only one diver, but there's always like multiple divers that do it at the same time. And you know, for example, on the first, it started in 2020. The person that did the productivity in Indo-Pacific, Renato Moraes, also came in this region to do it and to implement it here also with my supervisor. So yeah, there is a repeatability that has been tested. There is a control every year. Of course, it cannot be perfect once again.
No,
but
but I mean, there's always bias, but I was
it's trying to be
thinking if you
best
acknowledge
possible.
that and you incorporate that in your estimate, that's all. That's great. Perfectly clear. Thank you.
Thank you, Laura.
Yes, thanks for this very nice presentation. I wanted to ask you of the three fish that are dominant for the productivity.
Yes.
What they are all the same feeding the air here? They all feed on the same things? or
No,
not?
they don't. No,
It is,
after it was just right here.
yes.
So now, for example,
Oh, yes,
the main one is angular or linear. This one represents, as I say, like 35% of the productivity in more than 50% of the abundance. This one is the main one, but it doesn't feed only on invertebrate mobile feeders. It also feeds in plankton. That's why this fish is really important because it's categorized in invertebrate mobile feeder because it's his main food diet. But for example, also this one is an omnivorous and this one is an herbivore microherbivore. And on the 10 main species, I think is if I do it well, 6 of them are invertebrate mobile feeders, and 2 are omnivores and 2 are herbivores, detritivores, and herbivore microherbivores. So there is some, mainly between those 2 categories.
Because I'm assuming there is some relationship between their feeding regimes or the dominant feed, let's say, and the relationship with the resource that you measure in the benthic for those that are eating like the turf or whatever.
Yeah.
So did you have seen those trends or not? Like between what the fish eat and the relationship to the coverage of the benthic.
Yeah, we can see, for example, with omnivores and herbivores, we can see a decrease in turf coverage. So we can see actually that more there is herbivores, less there is turf.
And you see it with maybe I have not asked you properly, but these trends that you see, can you break it down at the species level to see?
We can, yeah. Because actually the great thing with productivity is that everything can be break down. Everything. If I want the productivity of only iron or atom, I can have it. If I want the productivity of only some fish or some transect or some years, everything can be done separately.
Yes.
And also, if I want only, I did also. It's not on the PowerPoint because I don't have time. But I did also the productivity and the gamma of the position in the water column. The one that stay on the bottom, the one that stay in the water column, the one that go out of the reef. And I did some.
Yes, of course you are. When you see those results, you are curious and want to pick.
Sure, and we can have a great conversation afterward if you want.
And then I have another question. You have an idea of the health of the reefs, like they were all healthy reefs. Have you observed?
Mainly yes.
Okay.
Is not those corals are more resistant to, for example, bleaching event. I think it's what you expect. Yeah. Because, for example, the turbidity of the water protect them from the radiation of the sun. This is one point. A paper that I cite is really amazing for that. It said that this ecosystem is really resistant, especially for bleaching event that will happen more often in the future due to climate change. And we see less bleaching event in this region due to those characteristics, like massive corals are more resistant. The water characteristics is also protecting them, and the fish associated are also, everything is related in the sense that this ecosystem is more resistant and resilient. Thanks.
Thank you, Lorenz. Good.
Thank you, yes. Well, parabéns. Congratulations. I do have a few questions or observations to have a conversation with you. Can you remind me what is the average size of a monoarolidiatum?
Actually, it's, I would say 15 centimeters.
Is it a species being catched for ornamental aquarium?
Actually, here we are in a marine protected area. So in this study, it doesn't include the fish and the fishing part and the ornamental catching. So it's really a preserved area. But I don't know, Orlinatum, I don't think is used in aquarium.
Yeah, but I know that this is a major problem in Brazil, and Brazil has many issues. We are very good on paper. We have many marine protected areas and so on, but the effective management and protection of this, it's not working so well everywhere. That's why I asked. But you were there, so if this is something that it was not commenting on the region of the study, then I assume that maybe it's not a big problem in that area of study.
In that area, actually, it's pretty well because when I was there, there was a boat for like tourists, and we asked them to leave if they were not with a guide. So tourists or tourists mainly are allowed there, but only with a guide that take care that they don't take anything from the area, they don't disturb. We cannot enter on the island. It's prohibited, and there is like venomous snakes in this also. So yes, this area is actually pretty well managed. Of course, we cannot be here everywhere every time.
Yeah,
but
then
generally it's a good one.
But then, what do you think then? What is the potential then of your study and data that you produce? Do you think that could it be used to stimulate protection of other areas where these species are occurring in a similar environmental context and are not so well protected, for instance?
I hope it will. It's our job to help the government to install this marine protected area, and I hope this paper will give them knowledge, and hopefully it will create other protected area. But I will not be there.
It's okay. One thing, if you have a little bit of time, can you go back to on your methodological session where you show the figure of your study site?
Here.
Yeah. Okay. Because I think I might have missed something. Can you just explain to me again? Your data comes from exactly which part?
So they come from ESSEC, Jardin de Corais, and Funio.
Okay. Did you in these explorations that you were referring to did you see any specific differences in the different areas?
Jardin de
Because they
Corais
look quite different, especially Funio and the other two.
Yeah, Funio is more protected because of the Jardin de Corais as the name says, there is a lot of. It's the area where we found the most corals there, and ESSEC, due to its position, is more. What is it? Exposed, exactly. It's more exposed. So we see differences, but on the productivity in general is not significant.
Oh.
There is a significant, but not among all the sites. There is a significance between Funil and ESSEC because there is the most exposure and the most protected one.
And what is the difference? Even if it was a trend, what was the difference? Do you remember? Okay. Because I think this is interesting. It should be a difference with the species. Maybe there's cryptic speciations of the species that are in Funil, for example, than in ESSEC. I would expect so. It could be part of the conversation.
Yes.
Just as a
I
suggestion.
can explore it a bit more for the paper. Thank you very much.
So, very few questions, Luana. Thank you for your presentation. You died.
Yes,
You
I
died.
did.
So you participated because you said that the sampling were in 2020.
Since
Since
2020, so every year since 2020, I participate to the dive of this year. Unfortunately, I'm not trained to estimate the species and size, so I didn't actually collect the data, but I was here to see how it was done. And I also participate to the data collection of the benthic coverage. Same, I wasn't actually collecting the data, but it was really interesting to see them doing it.
I was a little bit surprised about the difference in fish productivity between the two examples that you were putting in the Atlantic and the Pacific, because it was like 3,000 against 20. Or I was wrong.
Well, there's one side, I think there's 11,000.
Huh?
But the others are it's,
Actually, the Brajam Pat is 11 point something. Our in Southwestern Atlantic is 3.329. 3 point, while in Southwestern in the Pacific is 11 for the Brajam Pat, 4 point something for coral, the great coral area, and 3 for another point, and yeah, it was approximately in this range. So except Rajarampat is an amazing place with like really abundant fish.
We have no more time.
I'm
And
sorry,
you are
sorry.
done. And you are done. Thank you.
Thank you.
Thank you for attending this, and we see you in 30 minutes. Yes, thank you. Stop the
Good afternoon, everybody. I'm chairing this. Now we're gonna have three speakers, as you have seen in all of the other presentations. So we have Sarah now that's gonna be talking about stranding events in the Portuguese coast. So you have 15 minutes. I'm gonna just signal for you when it's 2 minutes left. And then if you go over, I'll probably stand up, but I won't like remove you, but I'd like you to finish by then, okay? Best of luck.
Thank you. Good afternoon, everyone. My name is Sarah Crawford. I'm here to present to you the temporal and spatial trends in stranding records of cetaceans and sea turtles along the southern Portuguese mainland coast, also known as the Algarve. My promoter was Professor Karim Ezzerini, and my supervisors were Dr. Ana Marcelo and Dr. Vivian Kalu. So first, I would like to talk to you about the stranding network, which was established in 2010 in the Algarve. It is a cost-effective and highly detailed method of data collection, as we are able to take both length and samples from individuals that strand on our coast. Unfortunately, this does mean that it is an underrepresentation of deaths, as we cannot track all the deaths on our coast. We are subject to the currents and the sea conditions to be able to access these deaths, and therefore it's also an opportunistic method of sampling. For our legislation, we have a couple of important laws that have been put in place to help protect our cetaceans and sea turtles. First is a decree law from 1981 in Portugal that prohibits the sale of any cetacean that was bycaught. In addition, we have an EU regulation from 2019 that means that there needs to be deterrents in the water to deter cetaceans from feeding in the same areas that we have fishing boats, and to try and use more ethical fishing here. And then also a directive from 1992 in the EU, which is a habitat conservation directive that also protects our sea turtles, which applies to Portugal. So in the Portuguese coast, we have many cetaceans that are found along our coast, and we have three main types of sea turtles. The aims and objectives of this project were to provide a baseline for the coast of Algarve using data from 1978 to 2023 for both cetaceans and sea turtles. This included the spatial temporal analysis of stranding events, including seasonality, so what seasons have the most amount of strandings, location, if there's any locations that are hotspots for these strandings, the morphology, including sex and size of cetaceans, and then size for sea turtles, and any main threats that we're seeing that are coming up in our results. For this work, it also is to provide insights into the relative importance of human interactions in our work, such as the activities of fisheries bycatch as a source of mortality. So first, I'd like to talk to you about our study area. This is our southern coast of Portugal, also known as the Algarve, and some morphological features that I'd like to point out. First is the western coast, which notably has high cliffs and bluffs, making this area a little bit difficult to access for our stranding network. And also the seas are more turbulent than our southern coast, which means that our strandings come and go at a quicker rate than along our southern coast. So they can be harder to access due to the time that they're spending there. Then I'd also like to point out the two lagoon areas that we have on our southern coast. In addition, we have an exceptionally narrow continental shelf at only 6 nautical miles, which means that that's bringing pelagic species closer to our shores. And we also have an estuary, which is the river that divides the Portuguese and Spanish border. So for our data collection, we collected the date, species, location, sex for only cetaceans, the length, the decomposition state, and cause of death for all of our strandings that were accessible through necropsies. And for our data analysis, I analyzed the data temporally by quarters, starting at the beginning of the year, such that January, February, March makes up 41, and then continues throughout the year. For our years, our periods are divided into periods of 5 years, except for our first period, which is a period of 6 years, just due to the number of years that we had. And spatially, I did kernel density or heat map analysis at a potential scale from 1 to 100, where it was interpreted linearly to 5 kilometers, with a spatial resolution of 0.5 kilometers. Length was used as a proxy for size class based on current literature, such that the individuals were divided between juveniles and adults. And the cause of death was first divided up into 3 categories for cetaceans, which is unidentified, natural, and anthropogenic or man-made causes. And for sea turtles, 2 causes, unidentified and anthropogenic. And then these causes were further subdivided where possible, such that anthropogenic was then divided into things such as bycatch, probable bycatch, trauma, and collision, and natural as well. So for our results, we have the trends of the pre and post-stranding network, our pre-stranding network in red, our post-stranding network in blue. And we can see a definite impact of the stranding network and the amount of samples that were able to be collected. This is not necessarily indicative of an increase in strandings in these most recent years. It's just indicative of, at the start of 2010, we can clearly see more strandings are being recorded, not necessarily that more cetaceans are stranded. We can also see this in the significance of our year periods, our red year periods having the most amount of strandings, our blue year periods having the least. And these last 3 year periods for both cetaceans and sea turtles were statistically significant compared to earlier periods. So we're having more strandings recorded in these year periods. So for our most common species that we're seeing stranded along the coast of Portugal, and some important details about them. Our most common species for cetaceans is our common dolphin. These are generally a coastal species. And then followed by our striped dolphin, our minke whale, our bottlenose dolphin, and our harbor porpoise, which are all pelagic species. Then we have, for sea turtles, our loggerhead sea turtle stranding more than our leatherback sea turtle. Some important details about loggerhead and leatherback sea turtles is that they are both pelagic or oceanic feeders and neritic feeders or coastal feeders. But these transitions happen for different times or for different causes based on these 2 species. For loggerhead sea turtles, this transition is happening due to size and as they age. They are, as juveniles, smaller and they are more oceanic, and then they become more coastal feeders as they get larger. In addition, they have a temperature, they struggle to regulate their temperature, and therefore when the temperature gets below 15 degrees, they become lethargic in these temperatures. For our leatherback sea turtles, they are transitioning from a pelagic or oceanic feeding to neritic feeding or coastal feeding based on the availability of prey. They're generally gelatinous feeders, but then they will transition based on food availability. And they don't have many struggles at regulating their temperature, so they don't become lethargic in cold temperatures, and in fact they're found in increasing sizes at the poles and smaller towards our equator. For our quarterly analysis, there were no significant differences found on 4 of our top 5 species. However, each of our cetacean species was found year-round, indicating a possible year-round presence for these species. And for minke whales, there was a statistical significance by quarter, specifically quarter 2 was statistically significant compared to other quarters, where we had more minke whale stranding in this quarter. Next, we had sea turtles, where there was significant differences between quarters for both loggerhead and leatherback sea turtles, such that loggerhead sea turtles were stranding more in quarter 2, and leatherback sea turtles were stranding more in quarter 3. And this could be due to several causes, such as seasonal temperature changes. For the distribution of strandings, we have an important note on this slide would be the lack of hotspots on our western coast, which could be, again, due to the morphological features that I mentioned earlier, such as our high bluffs and cliffs and a short time period that these strandings are staying on this coast. To go further into detail, our common dolphins are stranding in 4 hotspots here along our coast, and this can be correlated with our high levels of chlorophyll, specifically in this study by Faravetto et al. 2023, where we see visually a correlation between our hotspots and our chlorophyll A. For our striped dolphins and our bottlenose dolphins, our striped dolphins have some minor hotspots on our southern coast on this side, but more importantly, possibly, is our larger heat spots closer to our continental shelf, which is bringing, again, these pelagic species closer to our shores for both striped dolphins and bottlenose dolphins. For our minke whales, we have a notable increase in hotspots on this side of our coast. And for harbor purposes, we are seeing hotspots in our major estuary and lagoon areas. For both loggerhead and leatherback sea turtles, we're seeing hotspots closer to our coast. For loggerheads, they're a little bit further away from our shelf, but still close to this continental shelf. And for leatherbacks, they're very close to our continental shelf. So again, bringing these pelagic species closer to our shores. For size and sex trends, our striped dolphins had a significant sexual dimorphism, which is well studied, and our minke whale only had one adult recorded. All other minke whale strandings were juveniles, and it was found that it was statistically significant that more male juvenile minke whales were stranding than female juvenile minke whales. For our sea turtles, as we have no sex data available, we're just analyzing size. And for loggerheads, all of the loggerheads that stranded were juveniles, and for leatherbacks, 80% were juveniles. For a cause of death of cetaceans, I would like to draw your attention particularly on the side to the amount of dark blue that we are seeing, which is our anthropogenic causes, or our man-made causes. So a lot of our cetaceans are dying due to these man-made causes. So statistically significant here is in bold, and I'd first like to point out the fact that we have a statistical significance of trauma in our common dolphin strandings. So more common dolphins are dying statistically significant compared to other categories of trauma. We then have our other four cetaceans: the common dolphin, the minke whale, the striped dolphin, the bottlenose dolphin, and the harbor porpoise, which are all dying significantly due to bycatch. And lastly, we have the striped dolphin and our harbor porpoise, which are dying significantly due to undetermined causes. This could be due to the fact that necropsies are a surface evaluation of our animal, so we can see things like net impressions, teeth marks, these things to determine the cause of death, but we're not able to see things as clearly such as disease or other indicators of death that aren't very external or visual to the naked eye, indicating that we need more research for these two species in particular, as there could be high levels of disease in our population. For our sea turtles, again, we have high levels of anthropogenic causes of death. For our loggerhead sea turtles, our bycatch was significant. Notably, they are dying more due to gill nets. And for leatherbacks, bycatch was also significant, and undetermined causes of death were significant. So my thesis in overall underscores the importance of the stranding network in collecting data. We can see that our stranding networks have significantly increased the records or the size of our data availability in recent years and in recent years periods, especially since 2010. In addition, it highlights the anthropogenic causes of death, especially bycatch in cetaceans and sea turtles along the coast of the Algarve. And it also provides a baseline for cetacean and sea turtle research in the Algarve, so looking at areas that our species are stranding more and where they are stranding more seasonally and spatially. Further research is needed, obviously, into the causes of undetermined death for both cetaceans and sea turtles, as I previously mentioned. The fishing industry into individual understudy species, such as the minke whale, which doesn't necessarily have a cause for why we're seeing statistically significance that we are seeing. The fishing industry in the Algarve, so what exactly types of nets that we are seeing, specifically in cetaceans, which is not easily studied, and the oceanographic parameters such as chlorophyll A, sea surface temperature, and currents, as this is generally what brings these animals to our coast, and we're not necessarily able to, or there needs to be further research into this, that we're understanding exactly where these animals are coming from, such as Lagrangian models, and connecting these parameters to our strandings. Thank you for your attention, and I have bibliography.
Thanks very much, Sarah, for a nice presentation. We just open the floor for the public if they want to ask any questions. Yes, go on.
Your presentation was super clear. I just wondered, you said that juveniles of sea turtles were the main trend. Do you have a hypothesis on why juveniles are not adults?
So it varies based on species. For loggerheads, we are only having juveniles. There's a couple of different reasons. I again mentioned that they become lethargic at under 15 degrees Celsius. They're also transitioning when we have a narrow continental shelf into neritic feeding earlier, so they're entering a new environment, and this could be meaning that they're interacting with more things. This could also be due to the fact that we're close to a breeding area on the coast of Greece and Italy. So there are many reasons for loggerhead strandings that could be possible. For leatherbacks, I mentioned this gradient from our larger sea turtles at the poles and our smaller sea turtles at the equator, and that's likely due to the Sidus gradient that we have 80% of juvenile strandings for that species.
Anybody else in the public? No? Okay, so we're gonna share our floor here. Do you want to start?
Yeah, I have plenty of. Because, well, thank you for the presentation, really clear, really nice. You know, we started our stranded citizen network 1 year ago. And the thing is that it was interesting that you started in 2010, but there were some other data. What does data come from?
So that data comes from the Portuguese national government, and they have been recording strandings for longer than the stranding network has been available. In addition, in the north part of Portugal, the stranding network, I believe, was established in 2000. And so occasionally when we would have strandings and the maritime police would say that there's a stranding, if they had the time available, the northern Portuguese stranding network would come down and assist in those areas.
In Basque Country, that work was performed by the NGOs.
Yeah.
So sometimes that data is difficult to, as you perfectly well explained. And more technical things that, did you relate the degradation states of the
Yes, so the decomposition stage is very important because you can't really tell a cause of death at 4 or 5. Sorry, the decomposition state goes from a 1 to a 5. 1 is being very, very fresh. 5 is complete bones. And at the
Semami.
Yeah.
Bones.
At a 3, we can reliably or generally reliably detect the cause of death. At a 4 or a 5, sometimes we can, when you have things like straight cuts across fins, but those were discarded to keep the data more reliable.
It was nice to see that I don't know how accurate is the detection of the cause of the death, because around 50% that is noticed undetermined is a good number.
Yes, it's a high percentage, and again.
No. I mean, it's a good number. I mean, it's a lot of.
Yeah.
Because some papers say that normally it's around 27, 40.
So that could be for a couple of reasons. Our strandings that drift towards our coast have to be close to our coast, and so we're not getting the deaths that are just really far out at sea. But I would say that we're pretty good at detecting cause of death, essentially since we have such a large history of stranding networks in Portugal, and we have experts like Kareem to help us with fishing gear marks and such.
So you take more samples, and it's not only the necropsy external, and you
No,
take
so
samples.
we take samples for genetics, we take skin samples, we take teeth, we take
Histological.
yes, we also, there's a PhD in progress currently to look at discovering these things like viruses that could be present in our species.
Not only viruses, I mean, this is important information for the necropsy, or
Yes, there's many
pneumonia,
things.
whatever you can find.
You can also discover things like gas embolisms.
And then the samples
are stored in
They're
archive
stored.
in the bank.
Yes, they're stored in a bank. I don't know which bank. I have it in my paper, but I don't know what the bank is.
Okay, that's so interesting because we have the bank.
We do, but no one's touched it. So
Really
yeah, that's what that PhD aims to do.
makes sense. Yeah, it's good, really good. Very interesting. The last part that you mentioned, I think is the oceanographical parameters. We are nowadays doing quite the same idea in the Basque Country. And we are working also with physics, that is an adventure itself. I mean, yeah, physics. And we found a really nice relation between of course, the winds, but other specific events, atmospheric events. So
yeah,
it should be really nice because of the management of the network as well. So really nice, perfect.
Well, of course, I'm very familiar with your work, so nice job. But perhaps you could just briefly explain how the stranding network works. I mean, maybe the audience would be interested to know. Is it volunteers? Or how does it work?
So Stranding Network operates with a combination between fisheries, the public, and the maritime police. We eventually get a call or an email, usually with pictures, that says that we have a cetacean or a sea turtle that has washed up on our shores, and we have dedicated members of a team that are paid not very well, but we have two people and generally an intern as well that will go to that stranding and decide if it needs to be taken back to the lab or removed to a dump site. And then we will take photographs externally to look for things such as net marks, teeth marks that could indicate bycatch or interspecies interactions. And then we open up the animal to see what is inside or what could possibly be the cause of death. I know there's some people who helped participate in a necropsy in the room, and we found things like broken bones, we find nets in the stomachs of these dolphins, we find things wrapped around their tails. We find many interesting things when you open up a dead animal.
So looks like fishing activity is one of the main things, especially with gill nets and trammel nets. I was just wondering, do you have any comparison with what is happening in the north of Portugal? Because we know there's much more fishing effort with nets in the north than in the south. So do you think, are there more strandings per kilometer of coast in the north than in the south?
So there are more strandings. We get generally between 60 to 70 a year. Our maximum year was, I believe, 100, 110. And in the North Portugal, they get between 100 and 300 per year. So they operate many more strandings. And they also, their species composition is also a little bit different. They get many more harbor porpoises. Due to the fact that there are more harbor porpoises in the area, and we don't see as many harbor porpoise strandings. But that also could be indicative of different types of fishing as well.
You raise an interesting line of research, possibly including oceanographers and modelers, because, for example, when somebody is lost at sea, they often, the police call on physical oceanographers and modelers to look at the currents to see where the body could have showed up. Maybe here you could do the reverse, trace it back based on the age of the body and the currents, where it was maybe related to a particular fishery or something like that.
One of my colleagues here today, Patricia, did that in Ireland. She took strandings that were bycaught, and she traced them back based on the estimated day, based on the decomposition code, and did that. And I think that's a very important future study that needs to be done here as well.
So in our case, when the northwest wind hits, rise is it's like that. I mean, it's so clear. And mostly, most of the stranded ones are quite floating ones in that conditions because they are left possibly in the sea, and then they are, and it's really interesting those movements. And with the bycatch, and there are quite polemic nowadays in the major scale because they stopped some fishing nets because there were a lot of bycatch of tursios. So it's important.
Okay, I also have questions. So, first of all, yes, I may say, and I think in your thesis is also missing a little bit of that in terms of your methodology of the strandings, how are they, because we have to look at, we know that there's a lot of bias in stranding data, but we have to be able to spot where, these gaps are. You were talking about some hot spots there. Have you looked at anything to do with the topography of those areas? Or even the currents?
I have not looked at the currents or the topography. There's some idea that the continental shelf plays a major role, which I think I tried to emphasize, and the topography of the western coast being so difficult to access for our records. But for example, for our minke whale strandings, there's no research that has been done, including my research, that would explain why they're stranding mostly on this side of our coast.
So what was the sample size for the minke whales? In your presentation, you're missing sample sizes, like a bit more on the numerical.
Right. So it was 1,207 cetaceans that were sampled and over 800 sea turtles total. For common dolphins, I believe that was 300, and then it gets smaller as we go down. For the minke whales, I can't remember exactly, but I did have
because you had significant more males stranding, so that's why it's important to know your sample size for that.
Yeah.
And then just one last question. So can you tell me again your anthropogenic classification? Because it's a general thing, but you mentioned some smaller classifications.
Yeah, so in anthropogenic, it's broken down between bycatch, probable bycatch, trauma, and collision.
Okay, and where's litter in that?
We don't have any that have died of litter at all.
Okay, but you should include as a criteria. I mean, if you're finding like bits of fishing litter, that's litter as well.
Right. We've just had one that has stranded due to that, but in our data we didn't have any that ingested enough litter in my time period to be classified as a cause of death due to litter ingestion.
Zero data, still data definitely needs to be present because anthropogenic is the most important thing clearly in your data. Okay, that's all. Thanks very much and congratulations. So Mariana? Thank you. Okay, so let's welcome Mariana Barros. She is going to be talking to us a little bit about fish spawning aggregation in Honduras, Caribbean.
Thank you. So good afternoon to everybody. I'm Mariana Barros, and I'm going to present my thesis to you about developing a baseline assessment for 3 fish spawning aggregation sites in the Honduran Caribbean. Here.
Swipe with one finger. Okay. So what are fish spawning aggregations? So from now on, I will refer to them as FSAs. So this term refers to a grouping of fish of the same species in which they release their gametes.
These aggregations are characterized by significantly higher level fish densities compared to non-reproduction events. Usually in these FSAs, we can find the complex snapper groupers, but this study is about groupers. So groupers are top-level predators of coral reef ecosystems. They are a solitary species, but also a transient spanning aggregators, which means that they travel long distances to aggregate and span. And these aggregations are related with full moon. Furthermore, their presence is an indicator of coral reef health, such as in the Mesoamerican barrier reef. Okay. So the Mesoamerican barrier reef is an important ecosystem that extends from Mexico to Honduras. It supports a vast level of biodiversity essential for the formation of FSAs. Most of the studies about groupers and FSAs validated have been carried out in Belize, while in Honduras there's a lack of knowledge, and without this information, it's difficult to manage these aggregations.
You can probably also use the laptop if you
want. Oh, okay. So what is the problem? First, there's a lack of fish spanning aggregations validation. There's an indefinite ban for the Nasu grouper, but there's not budget for patrolling and compliance. In recent decades, also many FSAs have indicated signs of decline, and some of them have disappeared altogether. Despite this fact, these aggregations are still targeted. For that reason, the aim of my study is to validate the presence of fish spawning aggregations potentially occurring in Roatan, focusing on 4 grouper species: Pteroperca tigris, venenosa, bonacci, and Epinephrylus striatus. So Honduras is located in Central America, in the Caribbean Sea. It has 3 marine protected areas, 3 protected islands, the largest of which here is Roatan. So these are the 3 study sites: Manuar, Sandy Bay, and Cordelia Banks. These are the 4 study species: the tiger groupers, the black groupers, the yellowfin grouper, and the Nasu grouper that we can find in these 3 sites. These are important commercially and as a food source. These 3 species are protogynous hermaphrodite, which means that they are born as females and then become male. And usually females mature at the age between 2 or 5 years, it depends on the species, and then after 10 or 15 years, they start completing their transition to male. So we are talking about long-lived species. So what do we need to validate an FSA? So first is the visual verification of the aggregation, and then the spanning event. But literature mentions that it's difficult to observe these events, so they also use secondary indicators for spanning, which are color changes, courtship, grouping, also the observation of gravid females fighting, and bite wounds. So in the site, the Roatan Marine Park collected the data from 2021 to 2023, usually after sunset. In the site, the location was recorded, also the depth at the aggregation was observed, and surface and depth temperatures too. Also, the abundances were estimated. The species were recorded that were in the aggregation and their sizes. And finally, the fish behavior, which are the secondary indicators of spanning. For data processing, it was used R and also the tidyverse library. First, the peak day of the aggregations by species and site was determined. Then the behaviors only when spanning was observed. Also, the biomass was estimated using the growth parameters obtained from fish base. And finally, a heat map was made where the peak aggregation dates were in relationship with the lunar phases. So from now on, I will talk about results and discussion in the same slide. So for the depth, it was around 20 and 40 meters with a mean of 29 meters. With temperature, we didn't have so much data, but the interesting thing here is that the highest aggregations were at 26 and 26 26, and 26.6 for tiger groupers, which this aligns to this study that also they indicated that the tiger groupers span in this range that is between 24 and 26 degrees. So, are there FSAs in these sites? So first, I will talk about Cordelia bands. So here we can observe the individuals in the peak day aggregation, and here are the months. So first, we can observe that in Cordelia bands, there's the presence of the 4 species. By January in 2022, we observed that the tiger groupers and the yellowfin groupers exceeded 60 individuals. Despite the fact that no spanning was observed, the secondary indicators such as fighting and color changes were recorded. By March of 2023, nearly 100 individuals per species were counted. Here, the spanning event was recorded only for tiger groupers, but for the other 3 species, the full spectrum of all the secondary indicators were recorded. Here in April, we can see 2 peaks. One for the Nasu grouper with more than 150 individuals, and another for the yellowfin grouper with more than 200 individuals. And here, the spanning event was recorded for these 2 species and also for the tiger groupers. And for the black grouper, also the secondary indicators were recorded. So first, the spanning was observed for 3 species, and the secondary indicators were recorded for 4 of them, for the 4. So we can say that Cordelia bands are a multi-species spanning aggregation because it was recorded more than 1 species spanning in the same site. And also this aligns with these other studies in the Caribbean, in Mexico, in the Cayman Islands, and also in Belize. So here is Sandy Bay and Man of War. In Man of War, the studies started in 2022. So first, we can observe that in Sandy Bay, the tiger groupers were consistently the predominant species, recording the largest aggregation across the 3 years under analysis. The spanning event was observed in March and April of the 3 years. And in Man of War, despite the fact that these aggregations are smaller than in the other sites, the interesting thing here is that these peaks are in the same month as in Sandy Bay. So first, tiger groupers are the predominant species for these 2 sites. And this could be happening because tiger groupers are territorial. Also, another authors mentioned that tiger groupers have, when they reach their maximum capacity in the aggregation, some of them, because of this behavior that they are territorial, start going to other sites. And usually this range of the other site is approximately between 4 and 7 kilometers. So this could be happening between Sandy Bay and Man of War. Why? Because the distance between these 2 areas are 5 kilometers. And also here the spanning event was observed and also the secondary indicators were recorded. So here we have the behaviors when the spanning event was observed. In total there were 13 spanning events recorded on the peak aggregations days during the 3 years of the study. And here the interesting thing is that when we observe the spanning, also the color changes were recorded. So it's a good indicator of this event. Also the observation of rabbit females and the fighting, but in most cases the fighting was recorded for tiger groupers. Here we have a heat map illustrating the average number of groupers aggregated for spanning after the full moon across the different seasons between 2021 and 2023. Here are the species, and here are the sites. So why is this information important? Because Roatan Marine Park has a limited budget, so they have to choose some days to go and do the monitorings. So which are the best days to go and do these monitorings? So in Cordelia, we observed that the highest frequency is between the 4th and the 7th day. on the 6th day. And in Sandy Bay, the best days to go to do the monitorings are around the 5th, on the 5th and the 7th day. And in April, for Cordelia, we observed the best days are on the 7th and on the 8th days, and also the Danasio and the yellowfin groupers have the same pattern. And in Sandy Bay, we observed that the best days to go are on the between the 6th and the 11th day. And this also aligns with these studies that also have similar range of the fixed aggregations, and also to go and do these studies. And finally, the biomass was estimated. So here we observe the total grouper biomass recorded during the peak spanning aggregations each year, organized by site and species. So the only information that we can compare here are in Sandy Bay and Manowar, because we have the data available for the same month in the 3 different years. Here in 2. So what we can see is that there's a decrease of more than a half between 2021 and 2023, and also here is a decrease of approximately the half of the biomass. If we observe this graph, I don't know if you can see that, here in 2021, in March, for tiger groupers, they were recorded 200 individuals, and the next year was 250. So the individuals increased. But here we observed that the biomass decreased, so maybe that was because the fish that were observed were smaller. So conclusions. Sandy Bay exhibits a dominant tiger groupers aggregation where March and April are identified as the peak spanning season. Manowar is likely a spillover site from Sandy Bay once the primary site is at capacity. Cordelia Banks hosts a multi-species FSA for yellowfin, black tiger, and Nazi groupers. This study marks a significant milestone as it validates for the first time 2 groupers FSAs in Roatan, illuminating critical areas for groupers reproduction. And as next steps, I think that it's important to continue these monitorings in the 3 sites. Also, that is really important to record the temperature at the depth aggregations, so we can see and also study if there's any correlation between the aggregations and also the temperature. And it's important to extend this methodology and research to additional areas identified by features as aggregation sites. So here you can scan the literature, and thanks for your attention.
Thanks very much, Mariana, for a very, very good presentation. Does anyone in the audience have a question for her? Really? No? Okay, it's the last session and we start. Okay, so should we introduce? Yeah?
Okay. Yeah. Yes. So fishing on spawning aggregations, unfortunately, is one of the major causes of the depletion of long-lived, high-value species, and it happens all over the world, not just with groupers, but with the cyanids and high-value sea breams and stuff like that. So it's important to protect these spawning aggregations. I have a bunch of questions. One of them is why? I mean, did you know already that they were spawning aggregation sites? Because why did you choose those 3 sites? Was it known already?
Because there were another, there's more than 40 dive centers in Roatan, and the divers told this marine park that usually they see these aggregations, but they were not observed for the scientific group. So they started going. Also, they need funding. They have external funding that is from Coral Reef Alliance, and they started these studies in 2021, but they also have been, I think that they went to other sites that also the divers told them, but they were no aggregations. So usually they start because a group of people already observe these groups, and they it's like the start point.
What makes these places special? Why do these fish aggregate in these particular places? Is it a kind of bottom? Or what is it? The currents? Or what is it?
Usually it's because of the topography. Then you have all the corals that are the main habitat. Also you have mangroves that usually when they release their gametes and also the fertilized eggs, usually go to the seagrass and these mangrove sites, and they settle there. So I think that they can be a link between these places where they settle, like the larvae settle, and also the spawning aggregation. But there's not too many studies about this. It would be a hypothesis.
So you talked about numbers, and I was wondering why not density? I mean, you didn't give us any information on the methodology used in terms of the census, because I mean, okay, you may have 500 groupers in an area the size of a football field, but that's not the same as 500 groupers in this room. Mm-hmm. So density would be more, I think, more relevant than.
Yeah, usually.
Or are you saying that the divers covered the whole area, or?
No, it's because like usually when the groupers arrive to the site, like they stay between 10 and 12 days. So I was like also at the beginning thinking to work with the average, but in the end there are some things that arrive to the second day and they will be also in the peak. So I was going to double count them. So it's the reason I choose the peak day, and also in literature they work with this to do the comparisons. Yeah, I don't know if I answered your question.
Well, can I ask a couple more? Yeah. So these are, you said that some of them are multi-species spawning aggregations. Does that mean they're spawning at the same time?
No. It could happen, but usually a multi-species spawning aggregation is when a site is used for many species to spawn.
But not necessarily all
Not
together.
necessarily at the same time.
No, because I was wondering if there was any evidence of hybridization because they're spawning.
No, it's not necessary at the same time. It's that they use that area as a spawning site. Yes?
Okay, yeah. Thank you very much for the presentation. I appreciate as well when you, for the people that perhaps the audience is not accustomed to some of the terminology, abbreviations, and we can see. So the first slide, if I'm not wrong, was the explanation of FSA. Mm-hmm. So this is really nice, and I think it's also good for the audience to know that this helps a lot. Mm-hmm. Then to read the other things and to follow and to, so just a recommendation for all the people. Mm-hmm. From that point of view, formal aspect and also important for all people that this is supposed to be a master thesis. Mm-hmm. Which is your thesis?
It's about the validation of, first to see if there are these aggregations there, because like we have the data, but it was not.
No, but I mean thesis means conclusion. Mm-hmm. What is your conclusion of the work?
Like there are FSAs from Roatan, then there's also a multi-species spawning that should be protected. Also that there's
that there are a lot of conclusions.
Yeah.
this is because it's just the scientific methodology, and because sometimes we miss that this is some academic thesis is a conclusion, and if you have a thesis, you should have an hypothesis, and it's easy to relate. This is just because it's not my expertise, so it's easier to say that things and things. So it's clearly, but it's important also from time to time to say these things, I guess. You have clear what you have. It's not a problem. It's just the idea of some things we have to stick to a little bit better. Just to show that you have a really nice results and ideas. And together with that, what means validation?
Like you can know, like you can have that georeference of where is the aggregation. Because, for example, sometimes.
So, but this is validation of confirmation of what I mean.
Yeah, no, because sometimes fishers, also divers, can say that there's an aggregation in one, or there's not aggregation of a species in a place, and if you don't have, like, if you don't have the reference of where is the place, and also if you don't see any aggregation there or any behavior that this is occurring, you can't manage this space.
But maybe it's because depending on what we are working on, validation means other things for me.
Yeah.
Probably it's correct. I mean, it's nothing wrong, but it's more surprising because it's like you confirm that it happens.
Yeah.
You have evidence. I think that's what you mean.
Yeah, I think yes.
You have evidence, and it's good that you have evidence. I hope you publish that because it's important.
Yeah. Like next steps also are going to be to work also with the fishermen, because of course in a big group sometimes like you have a little group that want to do like conservation management, and other than they don't want, of course, that people know where is this aggregation so they can continue going and do illegal fishing. Yeah.
And the last and most important one. Did you participate in?
No, I mentioned in the methodology.
Yeah, but I think maybe next year, or maybe even if you don't obtain the data, but you have the opportunity to.
Yeah, I analyzed the information.
No, but I mean.
I went there.
Okay.
Yeah, I went there. And it was also, there was a problem because I was going to go for 3 weeks in February, but also there was not enough personnel to do the survey, so I have to change everything. And then I went to, in March, like 1 week, I went there. They trained me and everything, and I went to look for the aggregation, but that day we didn't have that. But it was really cool also. I think that people have to be really like dive masters to do these things, because the depth is like you can't see the bottom, and usually these spanning events are after sunset, so it's pretty dark. But it was cool.
Okay, thank you.
So just really quickly, Mariana, can you just like briefly summarize the methodology for those? I know that you didn't do them, but I feel like we're hearing bits of it, but not exactly how the collection of data was done.
Yeah, I'm going to go to that slide. Okay, so a group usually of 4 divers goes to the site, and then wait until 5, it's around 5 or 6 in the afternoon. So they go into the water, and they usually start looking at 30, 35 meters the aggregation. And then if they observe the aggregation, they try to estimate it, how many individuals are there, also to record the amount of species that are there, that if there's color changes, also you have to know about that, because there are some, for example here, some fish that change. This is a Nasu grouper that it was at the beginning of the presentation, that is like orange with lines like this. And right now you are watching. Now you are seeing a fish that is black and white. So sometimes it's difficult to record these events. Also sometimes they have cameras, also they can record it with a video. And when they have, they take notes of everything, and also the depth, the temperature sometimes, and then they process the data, like they put it everything in an Excel.
Okay. And is there like a protocol how they should behave and like not approach the group or stand, you know, a certain distance or anything like that? I mean, I suppose you don't want to disturb the event.
Yeah, usually is we have to be like 5 meters of distance of the aggregation. And of course, like 1 or 2 weeks before of the monitorings, people are trained because they work a lot with volunteers. They are 2 people that are in charge of these monitorings, but they have to work with volunteers. So they have to be trained first. And these trainings are also in the water. Like you go to the water, they tell you, like they show you like a fish or a school fish, and you have to take notes on it and everything, and then do the comparison in the boat.
Okay, thanks very much.
So, do I understand correctly that we changed to Rachel? No. Yes?
It's because we had supervisors in opposite time zones of the world, so it was better for New York to be in the afternoon and New Zealand to be in the morning.
Okay, so welcome, Rachel. So Rachel is going to be presenting. Had I read something about AI before? That's not there anymore.
No, AI.
So it was the other presentation that was just okay. So climate change, MPA effectiveness on small scale fisheries. Okay. Thanks very much. Good luck.
Thank you. And I'm gonna try not to fight with the technology too much. Okay, so like you mentioned, thank you. My name is Rachel Cohen, and I'll be presenting the influence of climate change and marine protected area effectiveness on small-scale fishery resources, and give perspectives for proper conservation. So first I'll give you a little bit of background on 4 key points that I think set the tone. So first, biodiversity. A high level of biodiversity, meaning multiple species that fill one niche, protect from climate change, anthropogenic change, even disease. And this protection creates a healthier ecosystem, and then if one of these species is weakened or removed, then there is an eutrophic cascade. So again, this is important to protect. And so small-scale fisheries, which is the data that I worked with, what they do, who they are is fishermen of the coast, and they're really important for industry because they provide lots of jobs, especially in Spain, and are culturally important to provide high-quality fish to the coastal communities in this area. The boats are quite small, between 4 and 12 meters, and they utilize coastal areas between 20 to 30 kilometers from the coast because they only do day trips. And these coastal areas are highly impacted by pollution and degradation of the habitat in this area. And because of these impacts, the fisheries resources can be declining, causing the fleet, especially in the Mediterranean where I did my study, to decline. And these fishermen, they have a seasonal rotation of gears, giving them the ability to fish multiple species, making sure, so therefore the biodiversity and being able to have many species within their landings is very important. So climate change, we all know that there's a lot happening in our world right now, but I'm going to focus on sea surface temperature. And this is an especially important impact to the Mediterranean because compared to the global average, it's
increasing in surface temperature 3 to 4 times global average. And this in turn is impacting the biodiversity and fisheries resources that are utilized. Tropicalization is a phenomenon that is currently occurring, and we're already seeing impacts of this, where the distribution and abundance of thermophilic species is increasing, especially in the Mediterranean, a semi-enclosed basin, again, warming much faster. No? Okay, we can go to that one, that's fine. Okay, marine protected areas, what are they? These are a management tool set in place to protect the ecosystem and especially that biodiversity and the resources. They have been proven to have the most effective are fully protected no-take areas, so no fishing is allowed. And the MPAs have increased in recent years, the number of them, especially due to our current UN Decade of Restoration, which is setting a global target to protect 30% of the ocean by 2030. So, as mentioned, my study area is the Mediterranean, specifically these two regions in the Spanish Western Mediterranean, Catalonia, and the Balearic Islands. And my general aims of this thesis are to evaluate the combined effect of sea surface temperature and marine protection and duration on the biodiversity and abundance of species. I have two hypotheses. The first being that the more protection and the longer duration is going to increase biodiversity, while the sea surface temperature is going to be detrimental to some species, and those thermophilic species, it will increase them. And the second is because I have two areas, I wanted to compare a more southern location, the Balearic Islands, thinking that it might have a higher biodiversity, higher abundance, and because of the influence of climate change, a higher turnover of species. So I was able to get my data from two different governing bodies, about 20 years for each area. And this was given to me in Davy data, so it was a lot. So instead I looked at the annual, I aggregated this to the annual landings and created a unit of landings per unit effort, landed kilograms divided by the number of annual trips. So I have lots of variables because I was able to combine all of these through a redundancy analysis, a multivariate analysis, which allows me to hopefully see the explained variation of the response variables by the explanatory variables. So I will go into a little bit of each variable in a second, but I just wanted to show you how in the RDA all of these variables were combined. So I ran 4 RDAs, 2 for each region, separated with the community and the species level. So I will talk about the community first, with the biodiversity indexes and total LPUE. So for talking first about the total LPUE, this is the annual LPUE per port. And like I said, I had a lot of data, so the Catalonia is in blue with 20 ports, and the Balearic Islands is 16 ports highlighted in yellow. As you can see, there's lots of variation between ports, and there were some areas that had significant trends over our study period. And if we look at the regional scale, we can see that in the Balearic Islands, here on the right, that there's a significant positive increase in LPUE, whereas in Catalonia there was the decline was not significant. And this was just to explain the species richness or observed species and my two biodiversity indicators of Shannon Index and Simpson Index, which was a combination of species richness and evenness. And we can see the results for Shannon in green and Simpson in purple that gave us in Catalonia a slightly higher variation over our time period of biodiversity, which was slightly different than my hypothesis. Next, I'll look at the species. So in total, I had a lot of species recorded, and in the Balearic Islands about 140, and Catalunya 240. So to minimize this, to make it a little bit more readable on the RDA and to be able to make assumptions and utilize information that was a little bit more reliable, I used the top regional species. So in each species is assigned a color, and each plot is a separate port. So in the Balearic Islands and Catalunya, basically what I want to show with these graphs is that there's a very high discrepancy between regions of the species caught. And even within regions, there is a slight variation of species. But we can go into this a little bit later. And lastly, I would like to talk briefly about my explanatory variables. So we have number of boats, sea surface temperature, the protection level of each port, and the duration of MPA. So, like I mentioned before, in the Mediterranean, the fleet of small-scale fisheries is decreasing, and we saw that trend reflected in our data. And I also saw the trend of sea surface temperature increasing, which was expected. And I'm going to step you through the process of assigning MPA index, and then how I shifted that to the port protection overall. So using Orca et al., which was a paper that designated an index level with the use of allowed fishing gears, the impact of these fishing gears, bottom exploitation, and the anchoring or boating use of the area, which combines the use and degradation of the area to create an index level 1 being fully protected and 8 being unprotected. And then with this index assigned to each MPA, I had 24 in total. I was able to assign a port protection index. And how I did this was with the 30-kilometer fishing radius, which I mentioned before was the area the fisheries used from the port. If an MPA fell within this area, then I was able to assign this port a protection level. However, if there was more than one MPA, it becomes a little bit more difficult. I used a weighted average and again assigned a protection level. And lastly, you'll notice an extra index level here, 9. If there was no MPAs within this fishing area, then it was assigned no close MPAs. And so this is a scale of our map with the fishing area around each port, and the polygons are each MPA with their protection level. And this is how it looks with the ports assigned their protection level. And then, just as an overview, I wanted to show the overall low protection in our area. We had a majority of that, and even in the recent years, which is shown in the second bar, that there was unfortunately not an increase in protection. And lastly, the duration. So whichever MPA within the fishing area was the oldest, that is how long I had the duration. Okay, so my 4 RDAs, the first being the Balearic Islands community, and then we'll go through all 4, and I'll try to pinpoint the most important aspects of each. So on the Balearic Islands, we have sea surface temperature highly influencing LPUE. And then you can notice that our biodiversity and species richness are all grouped together, which kind of makes sense between number of boats and high protection. So looking at this graph, this is the species, and hopefully it helps explain the main patterns. So sea surface temperature is affecting the positively influencing the spiny lobster and the dolphin fish. The dolphin fish being a more tropical species, and the fishery is increasing in the Balearic Islands. The biodiversity fell between number of boats and high protection, so this indicates that a majority of species fall within this protection, and it also highlights that the MPAs in the Balearic Islands are focused on promoting the biodiversity in the region. And lastly, I wanted to point out that the duration of MPAs is positively influencing long-lived species like the grouper. And so the last two. This is the Catalonia Community RDA, and you can see the biodiversity, like I showed before, is quite spread. And Simpson being more influenced by sea surface temperature and species by number of boats and low protection. And lastly, that the LPUE is influenced by low protection and number of boats. So the last plot that I have for you is the species of Catalonia. And we can see that the number of boats, sea surface temperature, and low protection also have a lot of the species represented here. And that sea surface temperature is positively influencing these two species that have literature to back up their increase. And lastly, that high protection is, this is slightly skewed, but sand eel and conger, high protection and duration. So this indicates that the MPAs in Catalonia are more focused on a single species fishery and not the entire promoting the biodiversity as a whole. So I would like to say my main conclusions, which are that all explanatory variables were significant in determining the biodiversity and the community, the species abundance. That sea surface temperature, we're already seeing effects on some species, especially the tropicalization phenomenon. That MPA protection in both areas was low, but that regionally they were affecting things differently, and that the species caught were due to cultural preferences, abundance, and differences in climate. And the main takeaways or advice for managers is that because of these differences in socioeconomic preferences and fish availability, regional management is advised instead of nationally. That the higher protection, because proven to be more effective, needs to be increased overall. And that because sea surface temperature has a large effect, the MPAs should reflect that and have that within their management plans in order to adapt and shift with the species that they are looking towards. And that lastly, that MPAs should have management tools to assess effectiveness. So I would like to thank all of my supervisors and the help that I got on this project. And thank you so much. Do you have any questions?
Thanks very much, Rachel. Well done. Do we have questions from the audience? Yes?
Sorry, maybe I missed it, but regarding the MPA protection levels, I was wondering how you assigned those numbers, or if you just retrieved them from a database, or...
A little bit of both. I didn't go into it so much because it was quite a length. Okay, this is now not working.
Go to the left side if you like.
I have a little bit extra. So, yes.
Nice.
Kareem probably knows a little more. He was on the paper that did this. But so first we there was 4 questions that were asked and a flow chart that we followed for each MPA. And within MPAs, there's zones, right? So each zone was assigned an index, and then the same like weighted average with the area was created. So you go through this flow chart, and you have the fishing gears have a level, and you choose the highest level of impact. And then you answer whether or not there is regulations on or the presence of aquaculture and exploitation, and then whether or not there are regulations on anchoring and boating, and then this leads you this final zone, and then you do the MPA. You go from zone to MPA, like integrating them with the weighted average as well.
Yeah, okay, thank you.
Yeah.
Anybody else in the audience? Oh.
very quiet in the room.
It's okay, it's the end of the day.
Okay, so do you want to go ahead?
Yeah, well, of course, I like the fact that you used our paper for your system.
Yes, I thought it was a good representation because we were using small scale fisheries and it was a major part of that index level. So.
Yeah, so the alternative would be the IUCN method, which doesn't take into account the impact of fishing behavior. So, anyway. I also like the fact that you used RDA. Most people use NNDS, but actually redundancy analysis is better because you can incorporate the explanatory variables and those arrows. Tell you.
Especially with the scaling that I used, it was the angle toward of each response variable to the explanatory variable is how closely they were influenced by it. So I found it very effective.
So obviously you had hundreds of big catch, many species. You said the top regional species, but what criteria was that? Accounting for how much of the total catch? Or
so the species that I showed accounted between 75 and higher % of the total catch, depending on the port, because some ports were very small and had limited data. But what I did was I took the species contribution percentage and I ranked that by region. So to find the regional species that had a commonality.
Yeah. In the first part, you used landings per unit effort, but that was for all the species, right? And not.
So the data that I got was daily data separated by species, because the landing data that I got is from the auctions. So every fish that's sold, they have a weight, a species code, and how much people paid for it, and the boat associated with it. And so it's I aggregated it a lot to get species by year, by port, and then the total LPUE was aggregated to the total.
Yeah, that's what I was getting at. That is a limitation of when you presented those graphs where you have the landings per unit effort for all those species. You lose a lot of information because some species there may be only one or two species accounting for a great in some ports.
That's why I tried to do the species percentage with the species graph.
The column,
Yeah. But because there were so many species to account for, I didn't think it was too messy to show you every species.
Of course, yes. Well, for now.
Just formal aspects. Put the graph where you have the slide that there are graphs everywhere.
there are many. This one?
That one.
Definitely.
Do you believe that I can read something?
Yes, I really liked this. So this is slightly different. Wait, this is not the exact one that I showed before. I was really worried about the animations not showing up. Can you tell? Here you go. This one's a little bit cleaner.
It's too much.
I agree, but I wanted to show the the work that went into it, but two, the differences between the regions and ports.
Yeah, but finally you don't comment that differences are that much because you are looking at the slide and say, shoop, and eliminate everything. Because I cannot understand anything, okay, there are some breath there, different than the other. You can present your explanation about all this data in other way, easier, or not.
Because I did everything on a port year level, I wanted to show that, but there are probably other ways I just didn't think of them.
It's for me that is not, well, for me. For people, I'm not, even I cannot see the years. I mean,
yeah.
I don't know, maybe some years are specific events that change something. One more, but I cannot.
Yes, 15 minutes is not enough.
Yes, but you have to know that you know that you have that time and you have to adapt that because you have a really nice data then to show, so maybe it's not necessary.
I appreciate that. Thank you.
You mentioned that biodiversity, which is important to protect. You say something like that. Really?
I believe so, yes, because especially seen in the Balearic Islands, you have the protection is protecting the biodiversity, and those fisheries are more sustainable. And so for the line of research that I did, it was important because the fisheries are more diverse.
If you were in a climate change, we are going to be able to protect something, or we have to adapt and it's okay?
Well, I think that was one of my main conclusions, was to adapt the MPAs to climate change scenarios, and this incorporates identifying what you want to protect, right? Because when biodiversity is being protected, you need to have different measures in place, whereas if you're doing a single species, more so in what Catalonia is doing, they are focused more on, like, these were not included, but they just open fishing restricted areas for trawling, but these only protect certain things. And you're seeing you're seeing more degradation along the Catalan coast than in the Balearic Islands, because the fishers are more involved and their fishery is like a higher percentage is more diverse, whereas in Catalonia you have a large percent made up of only a few species.
But I was just saying that because if you want to protect diversity, maybe we have to stop fishing.
That is one scenario, yes, but I don't think it's feasible, especially in coastal areas. Yes, so I think one of the ways that the Balearic Islands does this is they are very integrated with the fishers, and one of my supervisors is actually interviewing them and becoming, like, gaining trust and this kind of thing. And so then when you work more with the fishers to create protection, it allows them to feel like you're working for them and not against them.
And they understand that this is important.
Yeah.
And this is the critical point.
Yeah, so we're still getting slightly there. Like some of the fishers are on board, but obviously some of the ones that, back in my day, you know, they're not as on board. So yeah.
I believe that relation is really important, and I think in Basque Country, for instance, the Asti and the fishermen around, and it's true that the fishermen, they believe that what they say, the scientists, is like that, and you have to follow, and this is incredible.
Yeah.
Not all of them, of course.
Yeah, some of them think that if they speak with the scientists, that will take away all their fish, but we're trying to help improve.
The last one, if I can.
Yeah.
If you have to introduce more variables,
yes.
there is any that maybe should be really nice?
I
have one.
Well, I think that primary productivity, like chlorophyll, but also my supervisor is working with climate velocity, and this is like the movement of climate change and the fish like away from certain areas, and that is like kind of in our next steps is to incorporate that to see with the climate velocity where the MPAs should shift if the fish are moving in a certain direction, or so that is a future step. But I think yes, incorporating more environmental variables would be beneficial to see more impact.
I have one.
Yes.
Pollution.
Yes. Also.
And related with ports.
Yes. Yeah, we just didn't have that data available.
Yes, one.
Especially with the Spanish government, everything takes a bit longer.
Okay, thank you.
Thank you.
Okay, Rachel, can I just ask you a quick question just to summarize? So you were looking at small-scale fisheries and how the effectiveness of MPA protection was affecting them, and also how climate change is affecting them. So out of all of this data that you looked at, and you had a lot of conclusions, what do you think are the main things that we should take away from this, how they are influencing small-scale fisheries?
So first off, that marine protection is and can have a higher effect if the protection is higher. So I think that is a really important takeaway, because at the moment the majority is either not protected or unprotected, and that variables such as climate change, sea surface temperature are already affecting the fisheries resources, and so far that's not integrated in management plans and needs to be somehow integrated so that the shifts can occur when the species that they aim to protect are weakened or also shifting in location.
Okay. Thanks very much.
Thank you.
Thanks everyone for your attention, and yeah, enjoy the rest of the day.